Joining us as panelists are Andrea Leal, Executive Director, FKMCD. I'm Meredith Bensom, Oxitec's Head of Public Affairs and a Florida native. We have Kevin Gorman, Oxitec's Head of Field Operations, and Nathan Rose, Oxitec's Head of Regulatory Affairs. For tonight's agenda, we'll review how Oxitec manages pilot projects and data collection. We'll do an in-depth review of Oxitec's performance in the past. We'll also look at the Aedes aegypti Just Add Water technology optimization in Brazil. We'll review the proposed Oxitec FKMCD pilot project, and then we'll answer your questions. Now, I'll turn it over to Kevin to begin our presentation.
That's great. Thank you, Meredith. And thanks to everyone for taking the time to join. We're absolutely delighted that you could make it today. So I'm first going to take you really through some generic principles around the design of our field trials, and then through 8 different deployments that we've done over the years. And that includes the very first deployment, there was a performance trial, a suppression trial, if you like, through to the most recent second generation trial. Following that, as Meredith mentioned, Nathan and Andrea will continue on and talk about the Florida trial design. Just as a recap, I'd like to show it again because it distinguishes between first and second generation, and that's kind of important when we go through these trials because many of them are first generation and a couple are second generation, and that makes a little difference, as you'll see. So the first and second generation technologies, both self-limiting, they don't persist in the environment, and they have no long-term effects. They have a similarity in that they're both non-toxic and non-allergenic proteins, and there's no risk of indirect effects or direct effects on non-target species. But the second generation specifically is different because the phenotype, if you like, the effect only happens in females. Now, that means that it allows us it gives us the genetic sex separation that we can see in this strain. It's super useful because prior to release, previously, we used to have to mechanically sort everything for that first generation. And that was a very labor-intensive, costly process, lots of quality assurance attached to it. Now we don't need to do that. Now you can't produce females if you try because it's genetic, and we get 100% female lethality in those offspring. So that reduces our costs, but it super importantly reduces the complexity of the whole system. We have a more advanced fluorescent marker. I'll come into that in the next slide. And also, because our males survive, they're able to also pass the self-limiting gene onto their offspring, or at least to half of their offspring. And so we have a multi-generational pest suppression, which doesn't persist because it filters out or dies out in a half-life kind of fashion. So around about 5 to 10 generations, and after that, there's no persistence at all. That potential integration from those males that do survive allows us to potentially integrate our insecticide susceptibility, which is potentially a synergistic effect with insecticides. So it could help improve the potency of other chemical insecticides. And it should also potentially improve those chemicals, or at least sustain those chemicals and their effectiveness long term. And really importantly, and I'll come onto this now in the next slide, is the flexible deployment options that it provides us. So bottom left of this slide, you can see a box and a small capsule. And the fact that we can't produce females means that we can put eggs into a box, add water, and some diet for the developing larvae. And all we get is males that come out of that box. It transforms the approach from a very expensive, labor-intensive deployment system to something that's simple. It can be stored. It doesn't need special facilities, and it certainly doesn't need high-tech equipment to deploy it, or even very well-trained staff should it go through the trial stages. And on the right-hand side there, at the bottom, you see the fluorescent marker, the advanced marker that I mentioned. It's very bright. It's only visible under a certain wavelength, and it's apparent in larvae, pupae, and adults. And that's really important because it allows us to track and trace all the insects, whether they're released insects or whether they're offspring from released insects. And it's really critical to the surveillance system that we have, which allows us to see our performance and monitor our release rates. So there are three types of trials. Most fall into one of these three categories. The first is regulatory trials. These have a solid level of replication to give a certain statistical power. They're usually developed in close collaboration with the regulators because they have a specific remit to achieve a regulatory milestone. And most typically, for most countries, they look at biology or biosafety and efficacy. The second group is demonstration trials. These are sometimes larger, not always, but often a little bit larger than a regulatory trial, and typically have one treated site, maybe two, and they're more designed to show the difference in effect or performance of a particular product, and often that use an untreated control to make that difference visible. The third type are operational deployments. Oxitec have done some of those for its first generation, and those deployments tend to be looking at suppressing a population on a wider scale, and also are focused on the customer or the client or the collaborator's requests. So how do we collect the data? I mentioned the fluorescence, and that enables us to identify them in the last slide. Well, we use traps, a range of traps. On the left-hand side at the bottom there, you can see our egg collection ovary traps, the black plastic pot at the back. And this has a small substrate for catching eggs laid by females. And we use an app system to register each of those traps. And the second trap that we can use is an adult mosquito trap. This could be battery-powered, mains-powered, solar-powered, and it may have a counter on there which allows automated counting and uploading of the data through the cloud. And both of those traps are used to collect insects, and then we can sample them, decide back at the lab how many are Oxitec parented or Oxitec insects and how many are wild, and that allows us then to work out the exact performance over the course of a field trial. So how do we measure that performance? This slide is really to show you the 5 or so key metrics that we take, and they're in the table at the bottom. The chart at the top is really just to make sure everybody's clear on suppression. Suppression is a term that is used a lot for these kind of programs, not so much for chemical insecticides, but often for biologicals. People talk about suppression as a reduction in the population as a whole. And so in this sense, as you can see from the chart there, we have an untreated area in the blue bar and a treated area in the green bar. And it's really the difference between the two. And that's the level of suppression. And often that difference between the two also accounts for the differences that there were at the very start of the trial before the treatment began. It's a fairly simple equation, but I'm not showing it here because there really is no need. This visual description is far better. But you can see that it really is potentially through the course of a season going to fluctuate quite significantly. There could be a maximum suppression, there could be an average suppression, and that's why we get sometimes different metrics over different periods. And you know, the quantity of different numbers that can come out sometimes it can often be confusing. But that's suppression for you. In addition to that, we have abundance. So that's the number per trap, and those are the two green and blue lines that are used to work out suppression. That comes directly from the traps. There's direct counting of individuals. The overflooding ratio, that really is the number of fluorescent insects that we have compared to the number of non-fluorescent insects. We use males in this case because we release males, so we compare it with the number of wild males. And that allows us just to really see if we've got the dose rate correct or not. Then there's mating fraction. Mating fraction is the number or the percentage of individuals in the population that have been fathered by Oxitec males. And that allows us to tell us how much of the population we're treating. So if we're not treating enough, we can increase the number that we're releasing, and we can treat more of the population, and vice versa. Efficacy, that's the percentage of treated females that die. So if you treat a female, if a female is mated by a male and she lays some eggs of those offspring, what percentage of the females die? They're all treated, they're all been parented by an Oxitec male, and what percentage of those females die? And in 5034, we of course expect that to be 100%, and so that's really a great way of us confirming to the regulators that we have that level of mortality. Okay, so I'm going to go through now the field deployments themselves, a different slide on each of those, and as I mentioned, there's 8 to go through. The first one we'll cover in a second. I'll just quickly remind everybody that all trials have a regulatory background here. Regulators in all countries have certain stipulations, and every trial that we do is fully approved by regulators, and regulators typically also want to see a certain level of compliance through the trial, and we submit regular reports to those regulators as we go through them. These are the trials I'm going to cover today. As I mentioned, there's 8 of them, and then Andrea and Nathan will cover the one in yellow, which is the new proposed one. They do cover 4 countries I mentioned at the beginning. That's Grand Cayman, Panama, Brazil, and 8 trials, as I mentioned. 6 of them first generation trials with first generation mosquito, and 2 of them are second generation. So the first one, Panama. This was a trial, a demonstration trial back in 2014. It ran from April 2014 for 28 weeks through to the end of October that year. And our partner there was the Gorgas Institute for Health, a very credible research institute for human health with a long history going right back to the early days of mosquito control, in fact. We completed the trial in 28 weeks, a single season, as I mentioned, in a single area. There was 1 treated area, 2 untreated areas to compare it with, and we got 91% and 95% suppression against those different control areas. That was the maximum suppression achieved in that season. A key component of that trial was to show whether it had an effect on the local Aedes albopictus population, which was cohabiting in that particular area. And we showed that it didn't. We suppressed the Aedes aegypti population, and there was no effect, either up or down, on the Aedes albopictus population. 2 peer-reviewed publications came out of that collaboration, and we also demonstrated a very strong field mating performance. And one thing to note from that trial, not only was it very successful, but the public really warmed to the technology as we went through the trial. And late in the trial, when the vector control authorities came around to spray insecticides at one point, the residents asked them not to spray because they would kill the Mosquitoes amigalis, which is obviously the friendly mosquitoes, and that was where the phrase was coined. The next one is Oxitec trialing in India. Now, this has been a long-term collaboration since back in 2011. So it's in the early days, if you like, and it started both in the laboratory with mating studies with the local strains, and progressed through to 2016, where we had this very large cage suppression trial in these 6 meter by 6 meter cages. There's 6 of them actually on that platform that you can see in the picture, and they were opened by Soumya Swaminathan, who is now the chief scientist at World Health Organization. Those trials were regulatory trials, so they were replicated. Each of those 6 by 6 meter cages is split into 2 and is a paired treatment and control. There was 5 treatments and 5 control cages. They were then exposed to populations of Aedes aegypti in there. The local populations were exposed to Ox5138, this is first generation again, and monitored on a weekly basis over the next 34 weeks. And we achieved elimination, complete elimination of those populations within 15 weeks. So it was a very successful trial. It was replicated, and it really demonstrated for the first time that in a semi-field setting, population elimination with this technology was actually achievable. It also confirmed the mating ability, as I mentioned, with the local strains, and 3 peer-reviewed publications resulted from this collaboration. Cayman Islands, and we've had 4 projects in the Cayman Islands. I'm going to talk about 3 of them. The first one was really what some people would call a rangefinder or a biology study, and this next one, which is the first one I'm going to talk about, was the world's first GM mosquito performance trial. It's back in 2010, and really was the first proof of concept following the biology trial done the season before that showed that this kind of product could actually work to suppress populations. It lasted 23 weeks. It was basically a season, and it was a demonstration trial as opposed to regulatory. So there was a single site with untreated controls, and it looked at the level of suppression against those untreated controls, taking into account the differences at the start of the study. And up to 96% suppression was achieved within that trial, really validating the technology as a concept and underpinning all of what was to follow. So a very important milestone and a great partner in MRCU. The next trial in collaboration with the Mosquito Research and Control Unit, MRCU, in the Cayman Islands was not until 2016. Cayman Islands hadn't suffered any disease during that interim period until just before this, when dengue appeared on the island. Cayman Islands wanted to try the technology to see if it had any potential as an area-wide suppression tool. And so we did a relatively small project for around about 12 months with them to look at suppression within a particular couple of neighborhoods. The groundbreaking piece of this project really was the novel mobile laboratory that we built, shipped from the UK, and installed at the MRCU premises to complete the project. First generation was something that required a complex deployment, as I've mentioned, and having the right facility in the right place at the right time, it was critical to all of those projects when you're releasing males and you have the QC of those males to consider. So, you know, with a future vision, if you like, at the time, we thought, you know, we would try mobile laboratories, and this was our first deployment with one. The learning curve was steep, but we managed to achieve 62% suppression in that single season. And I would say that the Cayman Islands is one of the highest levels of Aedes aegypti that we've come across in all our trials. Some in Brazil too are very high, but the Cayman Islands have been extremely high. And so it was a pretty good result to achieve that 62%, and is certainly comparable, if not better, than what's achieved with most chemical strategies. So a demonstration trial again, rather than regulatory. So no replication, a single treated site and a controlled site. The next project in Cayman Islands was in 2018, and this also was a demonstration trial in West Bay. And in fact, looking at that satellite image there, far right, if you like, the top of the hammerhead, if you like, that is West Bay itself. This was a 22-week trial, again partnered with Mosquito Research and Control Unit, looking at area-wide suppression as a demonstration. And this time, in conjunction with MRCU, it was decided to look at an integrated approach. We knew that this wouldn't be a silver bullet technology, that it was about sustaining sustaining other tools and integrating other tools. And this was 513A as well, I should mention. This is the first generation. So a strategy was put in place to combine and rotate chemicals and Oxitec mosquitoes for this project. And we still used that mobile facility. Much of the operational niggles had been ironed out, and we had full compliance of the facility, but also in terms of operations, it worked very well. So all releases were at the threshold for females, and we made some great strides in terms of learning about how to integrate and combine these different approaches. Results for this in terms of suppression values are still being considered by the steering committee, and at some point, hopefully soon enough, those results will be released. There's a statement there from Dr. Jim McNally, who's the director at Mosquito Research and Control Unit, just reinforcing really how positive and productive the project and the operations were. This is Brazil, and this is an operational project, so a deployment of a different scale, if you like, in Brazil to the ones I've talked about so far. This 513A, so first generation operational deployment again, is was started with 5,000 people and worked up over several seasons to cover 65,000 people. So the majority of a decent-sized city outside Sao Paulo in Brazil. It's a very, a long-standing collaboration, a very solid partner in the vector control unit at Piracicaba, and really a great relationship built up over the years with the residents there also. We got 83% suppression or greater year on year for the 4 years that we were working in this area, and 98% suppression was the peak that we achieved. As I mentioned, it was 65,000 residents, and of those 65,000 residents, we polled over 1,000 independently through a third party, and 94% of those expressed a desire for the project to be extended across the city, and 93% expressed a desire for that project to continue as it was. So really great support by the residents after 4 years of ongoing projects, and a real sense of collaboration with the residents as well as the vector control unit. This next one in Brazil now moves us from the first generation into the second generation mosquito, or Aedes aegypti. So this is the first one that we did in 2018 with the second generation product. So this one is the one where the males survive. They can then give us extra suppression potentially, or they could potentially give us a better result sooner because they're released in a more effective way. This is a regulatory trial. It was replicated. There was 2 different doses. There was replication at each dose. It lasted 46 weeks. It was essentially a low season and a high mosquito season, and we got really great control right the way through the season. It was 96% peak, but super solid performance right from the start to the end of the season. It confirmed the biosafety credentials that the regulators are looking for, and contributed very strongly to the full approval for commercial release that we received from the regulators in Brazil. 94% of the residents, once polled after the project, wanted the project to continue, and as you can see from the statement there from the Secretary of Health of Indaiatuba, it was very successful and very positively received. So much so that we went back the following year with the second project, and this was our first project using the just add water approach. Same strain, but now we're releasing eggs as opposed to adults. This particular trial was super important because it was the first one trying the eggs, and It gave us a similar high level of suppression, of course, in small dengue-prone neighborhoods. But really importantly, it was shown to be faster than 513A. And we think that was due to the box, because those mosquitoes, those males are coming out when and where, when they want to in the daytime. When we're releasing adults, we're putting them out into the road, a particular time of our choosing around our operations. And it really isn't as sensitive as them coming out themselves into the environment in a much more discreet manner. So we got a really fast result here. It was 18 weeks for the whole trial, and within 15 weeks, we'd reached 92% suppression, over 90% in 14 weeks. So really a great vindication of the new technology, and showed that those egg release boxes were working really well with no females released, and of course, regulatory compliance. Okay, that's the 8 trials I wanted to cover, 8 deployments I wanted to cover in the different types. As I mentioned, 6 from the old generation, the first generation, and 2 from the new, the second generation. This is a list here of the field trials that have been published in peer review. I wanted to really just demonstrate to everybody that it's out there for you to read if you want to go and find it. Since we started doing field trials, we've published 7 of them from different field studies, a range of countries, and a total of over 100 publications in scientific literature. Since, nearly a decade now, we've been trying to do everything in open access, because that makes them free for everyone. And so whether you're a member of the public or an academic, you can find these publications online for free. And if you can't, then you can certainly go to oxitec.com/Florida, where links to most of them are available. So I think for a sort of final summary to this section, it's certainly the fact that performance of Oxitec mosquitoes has been super strong around the world, particularly when you consider it in the context of other control options that are available. We've published the novel findings. They're readily available, or there are novel findings such as that for the second generation that are in the process of review. And I think it's certainly true to say that 513A paved the way for 5034 success. Okay, I'm going to hand over now to Andrea, who's going to start the journey through the Florida Keys project. Thanks, Andrea.
Thanks, Kevin. Yeah, I really just wanted to, before Nathan gets into how the...
Andrea, I can't hear you, Andrea.
Back in 2009 and 2010, Key West had an outbreak of dengue fever. And you can see that in the graph. The highlighted and circled bars are the days or the years that we had those cases occurring in Monroe County. So, that really kind of got us thinking at the district, what can we do to supplement what we're already doing? How can we increase our control of Aedes aegypti specifically? And as you heard at that time, we had seen some of the initial information coming out of the Cayman Islands. So we really started looking at technology as well as others, really to combat and as proactive as possibly can to ensure that we're not seeing outbreaks. Unfortunately, we are currently experiencing another small outbreak in the Key Largo area this year. We do have 42 confirmed locally acquired cases, with a number of suspect cases still awaiting confirmation. So we're really looking for more tools to be able to help control this mosquito specifically. Another issue that we see in our mosquito population is that we are seeing insecticide resistance. So that really limits what we can do in response with the current tools that are out there. So I think, the bottom line is, whether it's Oxitec mosquitoes, whether it's Wolbachia or sterile insect technique, we are really looking at our district to add more tools to what we currently can do. We've seen that what we're currently using can suppress populations by about 50%. So really, we really need more more things, more tools to be able to control these mosquitoes. Another good thing as we're looking at any of this technology is, what is our environmental impact? Where we are here in the Keys, that's something that all of us are very concerned with. Obviously, we're surrounded by a coral reef system that's protected. There are multiple state and federally protected lands. So we want to make sure that what we're doing is in harmony with that environment. So looking at these tools, they are species specific. So only Aedes aegypti mosquitoes, doesn't affect any other species of mosquito. And so that is another, plus as we're looking at these technologies. And then also, during the regulatory process, all of these environmental agencies are weighing in. So looking at what they've come up with is extremely important in moving forward with anything. Nathan, over to you.
Okay, thanks, Andrea. I'm going to try and get through this fairly quickly to make sure we have enough time for questions. So looking at what the proposed pilot project is here in the context of what Kevin's been talking about, three main purposes. One, as Andrea's been saying, is to broaden the toolbox to help protect communities in the US against the invasive Aedes aegypti mosquito and the diseases that it's carrying. Second main aim is making sure that we can preserve both the quality of life for residents when it comes to protection from disease and also the preservation of the ecosystem. And then the third aim is to evaluate in the US context this safe, proven, innovative tool for fighting Aedes aegypti. So looking at what the project actually entails, four main components. First is community engagement, which we talked about extensively last week. And if you haven't seen that webinar, I encourage you to find it on our YouTube channel. The second is what we call a marker release recapture study. And then there are two projects, Project A, which looks at releases from a single point, and Project B, which looks at releases over a larger area. In total, we're looking at about 130 of these mosquito release boxes placed, about 28 weeks in the field, and 0 females will be released. Okay, so just going into each of those in a little bit more detail. The project design, as I've said, the single point release where we're trapping males and also the offspring using overtraps. Multiple point releases where we are really just focusing on the offspring of our male mosquitoes. We have replication and comparison to untreated controlled areas, as Kevin's described. And the specific locations where we will be carrying out these releases, if the project is approved, will be determined following monitoring of the wild Aedes aegypti population to make sure that we're in the right places. And timing, looking at a 2021 start, again, if the project is approved. The evaluation elements will be looking at a few different metrics. One is how far the males fly and how long they live. The second is how long does the effect of the males actually persist in the wild, what we call residual activity. We'll be looking at the ability of our males and the females that they mate to lay in natural breeding sites. And then we'll also be looking at the metrics that Kevin talked about, things like the percentage kill of female mosquitoes, which is efficacy, and the proportion of the wild population, which is actually being treated. All right. So two main parts of the project. Project A is a single point release, and this will be replicated up to 9 times. And that's where we place a single box and replenish that every week. And basically look at those metrics in that context. The Project B will look at a slightly larger area with multiple release points over that larger area. And as I've said, the locations would be selected together with FKMCD following pest monitoring and the initial community engagement. And for more information about community engagement, I do recommend our previous webinar, but I just wanted to highlight in this approach, there will be full coordination between FKMCD and Oxitec throughout the project, transparency and focus on robust information sharing, listening and learning from communities and other stakeholders, taking a broad view of who stakeholders are in this particular project. And as you'll be aware, there are multiple avenues for anyone to engage with FKMCD and with Oxitec as this project planning continues. Okay, in terms of timelines, briefly just to give an overview. So we would be looking at some work in sort of pre-mosquito season. including community engagement, staff training, and some pre-release mosquito monitoring to help us select the correct sites. And then the mosquito season itself would see the release of Oxitec mosquitoes in both these Project A and Project B scenarios, monitoring and evaluation throughout, using the various types of traps that Kevin's talked about. And then after the mosquito season, some follow-up post-release monitoring and obviously data analysis. And all of this is aimed at generating performance data of this mosquito in the field context to be able to allow regulators to assess how well this works in the US context. All right, with that, I will hand back to Meredith.
The first question this evening will go to Kevin. And this question is, and Andrea, if you'd like to also speak to this question, you absolutely can, but Kevin, we'll send it to you first. So how does Oxitec's performance measure up to chemical pesticides? What are the benefits of Oxitec over traditional methods?
Great question, and thanks for passing it on, Meredith. Certainly one that Andrea can chip in here, because Andrea's got more experience than anyone with using conventional tools. Certainly that's the case. In terms of Oxitec's performance, it is certainly pound for pound exceptionally effective, and insecticides, breeding site reductions, all education, all have their place in a vector control strategy, that's for sure. And so it's not really about, you know, which one's better than the other, it's about how to combine them in an overall approach. So there's a place for all of them, and certainly depends on the particular case. But Oxitec mosquitoes are very effective. They can give you percentage of performance, if you like, or suppression that often it's difficult to achieve with other technologies. And that might be due to an application difficulty. It could be due to insecticide resistance. It could be due to just the behavior of the mosquitoes, not necessarily the active ingredient in the insecticide. But the fact that our males go out and find the females, there's nothing better at finding a female mosquito than a male mosquito. When you've got, you know, a low population of mosquitoes out there, it's quite difficult to apply a chemical and hit them all with that chemical. But when you're putting out male mosquitoes to go and find them, that becomes much more effective typically. So when populations are low to medium levels, Oxitec mosquitoes tend to be very, very efficient. Andrea, maybe I'll pass to you.
Yeah, as I mentioned earlier, you know, we recently did a study, I want to say right after we started doing liquid larvicide from the air in Key West, and did a comparison between, you know, adding that into our arsenal versus, you know, conventional methods. And we saw about a 50% reduction in our Aedes-borne mosquitoes. So looking at conventional tools, they do have an effect on the population. But again, if you're seeing insecticide resistance, if we're seeing containers that are difficult to get to, whether it's by air or ground, you know, we're not going to have an effect. So I think that's why it's so important to look at novel tools to assist with that suppression.
Thank you, Andrea and Kevin. Our next question will go to Nathan. And the question is, has Oxitec considered doing a large cage trial in Florida? And if so, has it considered doing that first?
Thanks, Meredith, and thank you for that question. I think it's a good one. So the cage trial that Kevin described was in India, and India is actually quite unusual in that its regulators requested this as part of the regulatory process in that country. Most countries around the world don't have that requirement, and that includes the US. So in the same way as the US regulatory authorities, in this case EPA, regulates other mosquito control like Wolbachia technology, what they did is they assessed potential impacts on humans and the environment in permitting open field releases of the technology. And they followed that after very thorough evaluation of that strain in the lab setting. So that's already been done, extensive lab studies on the Oxitec mosquito. And in the case of the Oxitec mosquito, actually also, as Kevin said, 2 field seasons in Brazil already demonstrating safety as well as effectiveness of that particular mosquito. So when you have all of that background in your regulatory process, at least the way the EPA looks at that, there's certainly no need for there to be cage trials as an extra step in that process. So I hope that answers that question.
Thank you, Nathan. Our next question, Kevin is prepared to answer it, but Andrea, if you'd like to also answer the question, there may be space for both here. The question is, will the FKMCD staff be part of data evaluation? Who else will be involved in reviewing and evaluating the data?
Andrea, I'll just pass that one straight to you, I think. I'll pick up if need be.
Sure, and I can definitely answer on behalf of our staff. Yes, we will be involved, and we will be involved every step of the way. We all feel that it's very important for third party, which we consider ourselves to be, to oversee and evaluate data, collect data, identify traps, quality assurance. So we will be involved in every step along the way for sure. And then Kevin, I'll pass to you. You can talk about some of the other companies or organizations that are going to be involved as well.
Sure. So on top of FKMCD's independent review of the data and the operations, we'll also have an independent advisory panel for the project. And that's already been formed. It consists of a local businessman, veterinary surgeon Doug Mader. It also consists of representatives from the University of Florida and the Department of Health, who most people will have seen on various webinars, Georgie Ray and Bob Eady. So they form an independent advisory panel who are there to pass advice as locals, as US citizens, as experts in their own fields on all aspects of the project, not just data, but could be on all aspects. In addition to that, there's the CDC who have offered technical support, and that could extend from, again, operations right through to data evaluation to ensure that it's a nice, robust experiment. The regulators themselves obviously added to that. That's both at state level and at federal level, both FDACS, Florida Department of Agricultural and Consumer Services, and EPA, the Environmental Protection Agency, are keen to hear what happens with the project and will be reviewing the data closely.
Thanks very much, Andrea and Kevin. This next question is for Nathan. And the question is, do Oxitec mosquitoes persist in the environment? Do they pose any risk to endangered species here in the Keys?
Okay, thanks. So both of those are things that have been addressed by EPA in their environmental assessment of this application. So the Oxitec mosquitoes do not persist in the environment. They, as Kevin has said, the males can pass on their genes to their offspring, and the male offspring will survive. They can pass on the genes to half of their offspring, and so on. So those mosquitoes and the genes that they carry will disappear from the environment pretty quickly. And that has been demonstrated both in lab studies assessed by EPA and also in field studies in Brazil. So in the lab context, we would see those genes disappearing from the environment within 7 to 10 generations. In field studies in Brazil, we see that the 2 genes inserted into our mosquitoes disappear within a few weeks or a low number of months after releases stop. There may be some persistence of the background genetics in the wild population, but that's also something that EPA has assessed and determined that there's no risk to human animal health or anything in the environment as a result of that. In terms of endangered species, this is something which EPA assessed very carefully, and they made a no effect finding. So no effect on endangered species in the Keys as a result of the release of these mosquitoes.
Thank you, Nathan. The next question is for you too. And the question is, does the EPA agree with how you will review your data?
Okay, another good question. So the data metrics which Kevin talked about in the presentation earlier are things that have been pre-discussed and agreed with the EPA. So all of the details of those are available in the field trial protocol, which is available on the EPA's docket for this particular application. And there are links to that available through the Oxitec website. And you can see all of the details there that have been reviewed by the EPA. You can see their comments on that, and you can also see any additions that they may have made to the metrics proposed. So all of that is definitely agreed in advance with the regulators.
Thanks very much, Nathan. Our next question has a number of parts, and Andrea will probably ask you to answer the first pieces of it and then turn to Kevin for the remainders. The question is, and again, this is in several parts, resistance to organophosphate applications of pest control is a very real concern. Which chemical applications will be utilized during the experimental trials? What will the frequency of those applications be? How is that different from simply utilizing the chemical applications alone? And how will Oxitec determine the success of the experiment when chemical applications will continue throughout the experiment trial? If you need me to repeat, I can.
No, I can definitely handle the portion about what we'll be doing operationally while this trial is going on. And I think it's important to emphasize that we're interested in conducting these trials as a new, like a supplement into what we're already doing. So for us at Mosquito Control, we're really going to be business as usual, which means, we'll have inspectors in the areas. We will do larval control where we find mosquito larvae. We'll do adult control either by truck or aircraft if salt marsh numbers are very high, or if we're reaching thresholds of Aedes aegypti numbers. So the important thing is whatever we're doing in these treated areas, we're going to be doing in the areas that we're using as our control. So as long as mosquito control continues to do what it's been doing and doing so throughout all of these areas that we're monitoring, that really takes that factor out. So as far as what we're going to use, it's going to be the exact same things that we've been using because it's important that we continue to control salt marsh mosquitoes as well as the disease vectors.
Maybe I could just quickly add to that, Meredith. That's something, that's a situation that's always applied in all the trials that we've done. So local vector control authorities still carry on their usual control. Typically, in some places that's quite responsive as opposed to being proactive. So unless there's a problem, they don't spray. So often it doesn't actually go inside. But we can perform those trials. Note down, as specified by the regulators, both, the federal regulators, EPA, we will be noting down and submitting all the details of what has been applied during the trial, both to the control and the untreated and the treated areas, so that that's fully transparent and part of the independent assessment.
Thanks very much, Andrea and Kevin. This next question will also go to you, Kevin. And well, Andrea, and if you'd like to answer it too, that there may be space for both of you, but the question is, how will the data from the project be shared? Well,
it certainly will be a collaboration. So the data would be shared subject to steering committee approval in a scientific peer-reviewed journal. My personal opinion, although I'm not the editor of the journal that we submit to, is that it will be novel. It's the second generation, so it should be perfectly valid for publication in an academic scientific journal, and that will probably be the route, it's the most robust route for publication of scientific data following an item of research. So subject to steering committee approval, which is a collaborative effort between FKMCD and ourselves, it will go into scientific peer review journal.
Thanks very much, Kevin. Andrea, is there anything you'd like to add, or that may be all?
No, I think Kevin covered it, but it is extremely important for all of us as scientists to ensure that the data is put in publication as quickly as possible. So it's definitely something that we're going to work towards right away.
Great. Thank you, Andrea and Kevin. Okay, our next question, and this may be one of the last questions, but Nathan, this will go to you. And the question is, can you please explain again if there is any risk associated with tetracycline? Do the EPA, FDA, or state regulators think this is a risk of any type?
Okay, this is a question that we've definitely dealt with several times before. So I just want to say, if you do want a much more full answer, take a look especially at our second webinar, where we went through in quite a lot of detail as to how tetracycline is used in production in the UK. But that tetracycline will not be used in Florida. Mosquito eggs shipped to Florida will never have been in contact with tetracycline. Male mosquitoes that will actually be released if the project's approved will never have been in contact with tetracycline. So a lot of detail provided in webinar 2. But to answer the question head on, there is no risk associated here with the use of tetracycline. This is something that EPA has looked at. This is something that the Florida state regulators have looked at, including the Florida Department of Health. And this is something which the FDA looked at back in 2016 when they were assessing the first generation mosquito OX513A. And all of these looked at this exhaustively and found that there was no risk of spreading tetracycline-resistant bacteria as a result of the releases of Oxitec male mosquitoes. So really just to try and say very much head on here, no, there is no risk associated with this. And this is something which has been looked at now by 10 regulatory agencies in total.
Thank you, Nathan. We've got another question coming through now. And Andrea, this will go to you first, and then we'll turn to Kevin. The question is, have project plans been adjusted to reflect the FKMCD board's desire to see the project not interfere with COVID?
Yes, so I know that this was a major concern by our Board of Commissioners at our last meeting. And so we will incorporate a clause where COVID will be in consideration prior to any releases. This is also the reason that we're not even looking to do any releases until 2021. So yes, that is definitely something that will be taken into consideration prior to any releases.
I think we'll wrap it up now and just invite you all to visit our website. Oxitec.com/Florida is a webpage devoted to this proposed project. We do have frequently asked questions. Thank you again. Good night.